My name is Dimitri. I'm a scientist and a clinician, and I'm a chief scientific officer of the East Anglia Therapeutics, which is a spin-off of University of East Anglia. So we're working on developing new nanoparticle treatments for cancer. So a bit of a clinical need. I'm sure you know all this data that, in the Western world, 1 in 2 people will develop cancer, 1 in 6 men will develop prostate cancer, 1 in 8 women will develop breast cancer, and that's the 2 types of cancers we're really focusing on. Current chemotherapy is obviously not curative. We cannot administer significant amounts of toxic chemotherapy in the vein because we induce a whole body toxicity. So as a result, we give insufficient doses and the tumors develop chemoresistance. And overall, again, in the Western world, every 4th of us is going to die from cancer. So we're focusing on breast and prostate, the 2 most common cancers in women and men, respectively. The market is huge, and it's growing very rapidly, so from 136 in 2018 to 220 estimated in 2024, and $50 billion will be the chemotherapy market for breast and prostate cancers. Nanoparticles have been there for some time as a paradigm. They offer a few advantages, which could be tumor targeting through labeling nanoparticles with different things, toxicity reduction through binding the drugs into the nanoparticle and basically making them prodrugs and not releasing them in the circulation. And of course, potential co-delivery of multiple different therapies together, which is obviously a great idea because tumors develop multiple pathways of resistance. So targeting just one thing usually does not work very effectively. So how has it translated into clinic? Not very much at the moment. There are 7 nanoparticle drugs on the market. They're all single therapy nanoparticles, and all of those therapies are chemotherapies, not molecular targeted therapies. A very typical example is Abraxane, is a nanoparticle developed by Celgene. It's a paclitaxel chemotherapy bound to albumin, and then albumin is formed into a nanoparticle. Already generating $1 billion sales at the moment per year, and yet having only marginally increase in efficacy in comparison to the free drug because it's a single chemotherapy, so the tumors develop resistance eventually. So we approach it from a very different side. We obviously done a lot of research, and in many years we're looking at the molecular mechanisms of resistance. Proto-oncogene sphingosine kinase 1, SK1, is one of the potential targets for cancer therapy. It's upregulated in many tumors that is involved in chemoresistance, makes the cells resistant to particular chemotherapies. We found that there is a current inhibitor on the market called fingolimod. It's a drug sold by Novartis. It's a therapy for multiple sclerosis and never been used in cancer before. It's off patent as of August this year. And we found that giving fingolimod together with docetaxel therapy allows us to downregulate the mechanisms of cell resistance, which then allows the docetaxel actually to work and kill the cells more efficiently. More efficiently to the point that we can reduce 4-fold the effective concentration of docetaxel if we use it together with fingolimod. So far so good, but fingolimod has a very well-known side effect. It induces the reduction of the lymphocytes in circulation, so it cannot be used in cancer patients. And so we thought, imagine we have a targeting device which will bring it to the tumor and avoid all this toxicity. So hence our invention, the nanoparticle that is 100 nanometers in size, so it doesn't go anywhere else except the leaky tumor vessel. It doesn't go into a liver or kidney because it's too big. It's labeled with glucose, so it goes to the site of the high metabolism, and it carries fingolimod and docetaxel, which is bound to the polymer of the nanoparticle, so they are in prodrug form, not active in the circulation. We can add the MRI probe in there, so you can do both tumor treatment and imaging the same time. So a bit of preclinical data. So this is the characterization of nanoparticles, the scanning electron microscopy. They have all nice size distribution, and they release the drugs only in the acid pH, which would be inside the lysosome in the cells rather than outside in the body. At the neutral pH, they stay as a nanoparticle. And this is an experiment in mice injected with human tumors. we injected in the vein the nanoparticles, and they were circulating for a day, and then we have done the imaging. Nanoparticles were labeled with GFP, and you can see the tumors in green. If there was a bit of better color, you could see there is a little bit in the liver and the kidney and spleen, but none sort of not really comparable to what goes directly to the tumor. In terms of efficacy, obviously control does nothing. Fingolimod does not decrease the tumor's growth on its own. Empty nanoparticles are obviously not effective. Docetaxel has about 50% efficacy, and our nanoparticles are 35% better than docetaxel alone. Obviously, the combination of free drugs is actually even more effective in curing the tumor. However, if you look at the mouse weights, you can see that the free drug combination significantly decreases mice weight. They lose about 20% in the first 2 weeks of treatment, which is basically unacceptable toxicity. Nanoparticles carrying exactly the same combinations in the green circle cause no weight loss in this mice. Same thing about the white cells. You can see how the white cells are reduced by the free chemotherapy and not by the nanoparticles carrying exactly the same therapy. We compared it further in a more aggressive model of breast cancer, the 41 model, with anti-PD-1 antibody and Abraxane nanoparticles. As you can see in the red circles, both were not particularly effective, and actually half of those mice died, and we had to kill the rest of them. They were really moribund, while our nanoparticle, with the human equivalent doses of docetaxel and FTY, have been quite effective. So we have done patenting. We're currently developing a pipeline of these nanoparticles because this is not the only combination that you can use. You can use many different types of combinations. And we're actively collaborating with several pharmacological companies using their inhibitors in our nanoparticles. We have done the industrial scale manufacturing. So the Canadian company Precision Nano have manufactured these nanoparticles for us on an industrial scale, proving that this can be used further on in clinical trials. We have discussed this with MHRA, which we're really happy about this, because both drugs, fingolimod and docetaxel, are already for a long time used in humans, so we know all the doses, the responses, the toxicities, and pharmacokinetics. So they just asked us to do GLP studies, and then we can go to the phase 1, and that's basically what we're at. We are developing the GLP and then into the phase 1. And in terms of the work that we have to do, I think these are slides actually a bit old. I submitted a new version. So anyway, we have to do the GLP synthesis, which costs about £1 million. We have to do a bit of pharmacokinetics, pharmacodynamics, which is about 800,000, and a second species study for 700. But actually, we also include there a phase 1, which will cost another 3 million. So all in all cost will be £5.5 million that we need in this round of investment to complete this project within the next 30 months. So 18 months to bring it up to the clinic, and then 12 months for a phase 1. And we've got quite a team here. Our CEO is Tim Mustel, who has more than 20 years of drug development experience. I'm the CSO. We've got a chemist and a pharmacist on board. We're advised by James Spicer from King's College, who is the head of the phase 1 clinical trials unit there, and he's very happy to do a trial in his unit once we have achieved sort of the GLP milestone. And we're also collaborating with CPI, Precision Nanosystems, Fraunhofer in Germany, and Catapult and Covance are going to do the animal studies for us. So that's basically what we're going to. We're basically aiming to finish at the phase 1 with a $5.5 million investment, and we aim to exit or license this technology after the phase 1 to the big pharma, because obviously we're not big enough to make phase 2 and phase 3 ourselves. And of course, using that money, we will be testing and developing new combinations and new nanoparticles. We have already quite a lot of that. I just can't speak about it. They're not patented yet. We have quite a few de-risking steps as well. First of all individual components of the nanoparticles already used in human. So a big sort of step forward in terms of preclinical development. We already have agreed phase 1, so the oncologists are very happy with this combination and this approach. And upscale synthesis successfully performed, which again is a big thing in nanoparticle world. You have to prove that your nanoparticles can actually be synthesized on a large scale, not just in a little reactor in the lab. So overall chance of success are quite high for this early stage project. And again, our system has quite a few advantages over the current systems on the market, so either conventional chemotherapy, simple nanoparticles like Abraxane, or even fancy things like immune therapy in terms of targeting, efficacy, toxicity, and especially cost. If you compare it to immune therapies, our therapy will be much, much cheaper. So all in all, we have developed a new nanoparticle system for targeted delivery, increased efficacy, and low toxicity. It's a platform technology, can be used for many different drug combinations, which is really important because it's basically a way forward, and we can obviously test it in more solid tumors. And as a clinician, I think that this is tomorrow's gold standard therapy. If you think about the current standard, which has been developed in the 60s, the chemotherapy, after you develop some targeted treatments that are non-toxic and same time more effective, the patients will not accept being injected with the sort of toxic stuff anymore. And just as a little reminder, we all remember how the iPhone was launched in 2007. Apple did not have to invent neither the touchscreen technology nor the Edge or GPRS. What they've done, they've taken existing technologies and put it into a new package and just forwarded it to the right market and had a big success. And that's what we're aiming to do with our nanoparticles.